Welcome to this lecture on the biochemistry of metabolic processes. Today, we are going to explore a highly crucial topic known as deamination. If you have ever wondered how your body safely processes the proteins you eat without poisoning itself, you are in the right place. Let us dive into the fascinating mechanisms that keep our cells functioning and our bodies alive. To begin our journey, imagine a car that you want to recycle for its valuable metal frame, but it has a highly toxic and explosive battery inside. Before you can safely melt down the metal frame, you absolutely must remove that dangerous battery. In our bodies, amino acids are exactly like these cars. The metal frame is the carbon skeleton, which is incredibly useful for making energy. The toxic battery is the nitrogen-containing amino group attached to it. We cannot just burn the amino acid for energy while the nitrogen is still attached. Therefore, the body uses a dedicated dismantling step called deamination. Now that we understand the analogy, let us look at the actual biology of this dismantling step. Deamination is the biochemical process where the amino group is completely removed from an amino acid. Unlike transamination, which just moves the group around, deamination completely liberates it as free ammonia. In the watery environment of the cell, this ammonia quickly picks up a proton to become an ammonium ion. The most common form of this is oxidative deamination, happening primarily in the liver and kidneys. Once the nitrogen is stripped away, we are left with a non-nitrogenous carbon skeleton called an alpha keto acid, which is now perfectly safe to use for energy. Understanding how this process works leads us to ask why it is so absolutely critical for our survival. The human body has no storage mechanism for excess amino acids, unlike how we store fat or glucose. When we consume a high-protein diet, the excess must be broken down and repurposed immediately. Deamination provides the only safe pathway to separate the useful carbon skeletons from the potentially lethal nitrogen. If this process were to fail, ammonia would rapidly build up in the blood. This condition, known as hyperammonemia, is highly toxic to the central nervous system and can quickly lead to severe brain damage, coma, and even death. Since we know the stakes are high, let us examine the exact chemical equation that makes this safe processing possible. This formula represents oxidative deamination, where glutamate acts as the central collection point for nitrogen. NAD+ is an electron carrier, essentially a biological taxicab waiting to pick up high-energy electrons. Water is used to hydrolyze the molecule. On the product side, we get alpha-ketoglutarate, which is the safe carbon skeleton. NADH is the taxicab now fully loaded with electrons, heading to the electron transport chain. Finally, we produce the ammonium ion, the toxic battery that will be sent to the urea cycle. If glutamate levels increase, the reaction pushes forward, producing more ammonia and energy. To truly grasp this reaction, let us look at a structural diagram of what is happening at the molecular level. On the left, you can see the structure of glutamate. Notice the nitrogen-containing amino group, which is the toxic portion we need to remove. The central arrow represents the enzyme glutamate dehydrogenase, driving the reaction forward. You can see NAD+ and water entering the reaction, while NADH, a free proton, and the toxic ammonium ion exit. On the right, we are left with alpha-ketoglutarate. The amino group has been completely replaced by an oxygen atom, leaving a clean carbon skeleton ready for energy production. Now that we have seen the chemical structure, Let us quantify the energy this process generates. This simple formula comes directly from the stoichiometry of our chemical equation. It tells us how many electron carriers are produced based on the amount of starting material. NADH represents the total number of NADH molecules produced. N-glu represents the starting number of glutamate molecules. Because the chemical ratio is exactly 1:1, we simply multiply the starting amount by the constant 1. If the starting amount of glutamate increases, the production of these high-energy electron carriers will increase in direct proportion. Before we move on to total energy, let us see this formula in action with a concrete example. Suppose a liver cell processes 10 molecules of glutamate during a period of fasting. We are given that N-glu equals 10. We choose our stoichiometric formula because it directly links our reactant to our desired product. By substituting 10 into the equation, we multiply 10 by 1. The final answer is exactly 10 molecules of NADH. Physically, this means the cell has successfully loaded 10 biological taxicabs with high-energy electrons, which will now travel to the inner mitochondrial membrane to power ATP synthesis. Having calculated our electron carriers, we can now determine the actual usable energy generated. This formula converts the intermediate NADH molecules into ATP, the primary energy currency of the cell. eATP represents the total energy yield in the form of ATP molecules. We take our number of NADH molecules and multiply it by the biological conversion factor of 2.5 ATP per NADH. If the efficiency of the electron transport chain were to decrease due to a metabolic issue, this conversion factor would drop, resulting in less total energy for the cell. Let us continue our previous example to find the final energy yield. We know from our last calculation that we have 10 molecules of NADH. We need to find the total ATP, so we use our energy conversion formula. We substitute the 10 NADH molecules into the equation and multiply by 2.5. The final answer is 25 molecules of ATP. This physically means that just by removing the toxic nitrogen from 10 amino acids, the cell has already harvested a significant amount of usable energy, even before burning the leftover carbon skeletons. While harvesting energy is great, we must also manage the toxic waste produced. To quantify this waste, we first need to know its molar mass. This formula calculates the molar mass of a single ammonium ion by adding the atomic masses of its constituent atoms. MNH4+ is the total molar mass we want to find. Mn represents the molar mass of nitrogen, and MH represents the molar mass of hydrogen. We multiply the hydrogen mass by 4 because there are exactly 4 hydrogen atoms attached to the central nitrogen atom. Since atomic masses are constant, this value will never change. Let us calculate this constant conversion factor so we can use it in clinical scenarios. We are given the standard atomic masses, 14 grams per mole for nitrogen and 1 gram per mole for hydrogen. We use the molar mass formula to combine these individual weights. Substituting the values, we add 14 to the product of 4 times 1. The final answer is exactly 18 grams per mole. This is the critical conversion factor we will use to translate the chemical amount of toxic waste, measured in moles, into a physical weight, measured in grams, which is essential for monitoring patients. Now that we have our conversion factor, we can calculate the actual physical burden on the liver. This formula translates the chemical amount of substance into a tangible physical weight. The variable m represents the total mass of the ammonium produced. The variable N is the amount of substance processed, measured in millimoles. We multiply this by the molar mass we just calculated. If a patient consumes even more protein, the variable N will increase, leading to a directly proportional increase in the toxic mass produced that the liver must handle. Let us apply this to a real medical scenario. A patient on a very high-protein diet rapidly deaminates 50 millimoles of glutamate. We need to find the physical mass of the toxic byproduct. Because of the 1-to-1 ratio, 50 millimoles of glutamate produces exactly 50 millimoles of ammonium, so N equals 50. We substitute 50 and our molar mass of 18 into the formula. The final answer is 900 milligrams. This physically means the patient's liver has just generated nearly a full gram of a highly toxic substance, which it must now immediately convert into safe urea to prevent severe neurological damage. We have covered a lot of ground today, so let us review the most important ideas. First, deamination is the essential metabolic gateway that allows our bodies to safely utilize proteins for energy. By carefully stripping away the nitrogenous amino group, the cell separates the toxic waste from the valuable carbon skeleton. Second, this process is not just about waste removal. It yields immediate energy in the form of NADH. Finally, understanding this delicate balance is key to knowing how our bodies manage high-protein diets without poisoning themselves. You now have a solid understanding of this vital biochemical process.